Hello. I didn't know this was not recording. Okay. So I did my presentation on flow cytometry. So flow cytometry is the process by analyzing the physical and chemical characteristics of a particle in a fluid, primarily in diseases or disorders that affect the blood. So the blood is a fluid, and there's cells that reside in the fluid. So the fluid passes through the laser, and the cells in the fluid are fluorescently labeled and then released by the laser to emit light at varying wavelengths. So that will allow you to basically organize and characterize the cells in the blood based on their wavelengths. And this is usually done in the field of immunophenotyping. So basically looking at the immune system and what cells are involved in it. Flow cytometry process is usually performed in cell counting, cell sorting, detection of biomarkers, and protein engineering. So cell counting, basically, if a patient comes in and they have a low blood count or a low white blood cell count, flow cytometry can help with that, can basically tell you, okay, this is the amount of red blood cells you have, these are the amount of white blood cells you have, etc. Cell sorting, pretty much the same thing as cell counting, but allowing you to sort the cells based on how they look. So, for example, if someone has a blood disease or something, um, flow cytometry can organize the healthy blood cells, which will be in a circle, in a nice round circle shape, whereas if someone has sickle cell, there will be some blood cells that are circle, round, and plump, and then there'll be some cells that are like in a half crescent moon. So flow cytometry can sort that out. Detection of biomarkers, basically, if there's any receptors on the cell, the flow cytometry can detect that. Protein engineering. I'm not really too sure about this, but in the discussion post, we can definitely discuss more about it. So there is an article titled Primary Immunodeficiency Diseases Using Flow Cytometry. Basically, what the researchers did was they wanted to look into whether flow cytometry can be used in primary immunodeficiencies, which are heterogeneous group of inherited disease of the immune system. So basically, they want to see if flow cytometry can basically organize these diseases because they do have some similarities. So flow cytometry can evaluate specific cell populations or subpopulations, cell surface, and intracellular and intranuclear proteins, which is really helpful in primary immunodeficiencies. So these are the the diseases that the researchers tested, and they wanted to see if flow cytometry can be done with patients who cannot really afford great health care. So these are the diseases the researchers looked into, and this is an example of the results. So this is the patient T cells, B cells, and NK cells um, were separated by based on the lymphocyte gate. T and NK cells are absent, and the number of B cells are increased. So right here is the dark spots are the B cells. So basically, what happened was they separated the TB and NK cells, and they saw that the B cells were very high. So further research, the researchers did quite a great job because they looked into different diseases under the umbrella of primary immunodeficiencies. Um, but however, since they wanted to look at whether this can be done in a low cost area, they didn't include where they can possibly do this. Especially, my assumption would be like areas where there's high poverty, and then also how much this could cost. So I feel like if this article was to have like a part two or further research was done, then they would look into where exactly this can be performed at and how much. One question I like to look at myself is can a knockout method be used in this application? Because there are some cells and there can be some mutations that will cause the cells to either act the same way as a healthy cell or could be an affected cell but doesn't phenotypically look like a affected cell. So possibly looking to whether a knockout method can be used in this in flow cytometry. So here are my references. If there's any questions, concerns, sorry, no, not concerns. If there's any questions, we can definitely discuss them in the discussion post. Any critiques that will help with this presentation to be better. Thank you.